Hey guys, this is Mr. O'Brien here, and this is your Topic 3 review video for the Keystone Biology Exam that's coming up at the end of the year. Now, Topic 3, we're talking about the cell, its organelles, and how cells move things throughout the cell. So let's start out with the cell theory. So the cell theory states that all living things are made of cells, okay? Now, all living things are made of cells. What they're talking about here is that every living thing, every single one of them is composed of this single unit called the cell, which is why the cells are the basic unit of structure and function in all living things. So for example, like, plants. Plants have cells as their base for every living thing. Same thing with animals. Same thing with plants. Bacteria, all of them are made of cells. And then finally, the third thing is that new cells are produced from pre-existing cells. Now, this is this idea of biogenesis. And biogenesis is this idea that living things come from other living things. Now, originally, people thought that biogenesis was this really far-fetched idea, and they believed on something called spontaneous generation, where they came from others. They just appeared out of thin air. So the first guy that kind of came up with or saw cells was a guy named Robert Hooke. Now, Robert Hooke, he coined the term cells, and why he called them cells is he thought that they looked like the rooms that monks lived in. Now, there were a couple other guys. Anton von Leeuwenhoek was the first guy to observe living cells, and he looked at his teeth scrapings. And then we had Schleiden and Schwann, who came up with the idea that all animals and all plants are made of cells. So all these people together, they came up with this idea of the cell theory. So two different types of cells that we have. We have prokaryotes and we have eukaryotes. So prokaryotes are single-cell organisms that lack a nucleus and organelle. Now, the key distinguishing characteristic they have is that they have no nucleus, or they lack a nucleus as well as organelles. So they also have circular DNA, and bacteria is an example of this. So a prokaryotic cell is pretty simple. Okay, you think of a bacteria. Now, inside of that, they have circular DNA, okay, and that DNA just kind of floating in the middle, not in a nucleus. Now, they do have ribosomes, which is where our proteins are made. And what they also have are these little hair-like structures. Now, they could be numerous and very small that go around the entire thing, or there could be just one big one. Now, these little guys right here, these are called cilia. And cilia are just little tiny hair-like structures that aid in movement. And this big guy are called flagella. Again, they move in a whip-like pattern. So again, prokaryotes, very simple. Okay, bacteria is the example that we have here. Okay, and this is believed to be the first type of cell that actually evolved because of how simple they actually are. Now, eukaryotic cells, they are distinguished by containing a nucleus as well as organelles, so membrane-bound organelles. They are much more complex, and plant and animals are examples of our plant and animal cells. So our eukaryotic cell is much more complex. Now, inside of a eukaryotic cell, we have our nucleus, okay? And this nucleus is where our DNA is contained. Now, it is linear in shape, and there's enough DNA in humans actually that in one cell, there's enough DNA to stretch 6 feet. Now, inside of these things, we have other organelles, like the Endoplasmic Reticulum or the Golgi bodies, okay? Now, we also have ribosomes as well located around, We also have mitochondria, which are the powerhouse of the cell or energy. So again, much more complex, okay? You can see there's a lot more organelles, a lot more membrane-bound objects on the inside of that, okay? So all cells are enclosed by these things called a cell membrane, just the outside covering of the cell, okay? And within a membrane is a nucleus, which stores our DNA in eukaryotic cells, but in both eukaryotic and prokaryotic, there's cytoplasm, which is just almost like a liquidy substance that holds everything in place. So within the cytoplasm are organized structures that perform a task called organelles. So again, all these little things right here, these are organelles inside the eukaryotic cell. Now, in the prokaryotic cell, we don't have those type of things. We just have, a DNA region. So inside, there's a bunch of different organelles. The first one we have is a cell membrane. And a cell membrane, all it does is it surrounds the outside of the cell, okay? It allows certain things to come in and certain things to leave. It's what's known as semi-permeable, okay? And semi-permeable just means that certain things are able to come in and out and leave the cell, okay? So it stops certain things, other things it says come on in, okay? So it's composed of what's known as a phospholipid. And you often hear this as the phospholipid bilayer. And the reason for this is phospholipids are arranged, okay, into layers, okay? So they kind of look something like this. Now, the heads are hydrophilic. And what that means is that they're water-loving, or they like to be associated with water. The tails are hydrophobic, okay? They don't like to be associated with water, which is why they associate on the inside like what you see there. So, next organelle that we have is the nucleus. And the nucleus right here is where our DNA is stored. So it contains all of the hereditary information inside of the cell. So DNA is the structure that most cells use as that hereditary information, okay? So again, our nucleus contains the genetic information. Now inside of that nucleus, you'll notice if this is our nucleus, there's this region on the inside that is tightly coiled. That is actually called the nucleolus. And the nucleolus is actually where our ribosomes are made. So, okay, we have a nucleus which has our genetic information. Now, right inside of that, there's a tightly coiled region called the nucleolus. Our ribosomes are where proteins are made, okay? Protein synthesis, they're like the factory of the cell, okay? So they allow certain things to form. So if you look up here, you'll notice that we have our proteins budding off of these ribosomes. So ribosomes are located in two locations. They're located in the cytoplasm, and they are also located on the rough endoplasmic reticulum, or just rough ER. Okay, so again, this is the site of protein synthesis, where our proteins are made. Now the mitochondria, you'll often hear this as the powerhouse of the cell. So the powerhouse of the cell is where ATP is made. So, site of ATP is produced during cellular respiration, okay? During cellular respiration, this is the organelle that is responsible for doing all those tasks. Our chloroplast, this is the site of photosynthesis in the plant cell, okay? So, in other words, we're going to take sunlight, we're going to take carbon dioxide, and we're going to make glucose in our cell. So, this is our other factory molecule inside of our cell, okay? Now, a vacuole stores things, okay? So in plants, we call this the central vacuole, okay, because of how big it is. In animals, it's not nearly as big. It's just a simple storage mechanism, a lot smaller. So the vacuole stores water, undigested food, and or waste. Very important in plant cells for storing all this matter. So we also have the endoplasmic reticulum, and what this does, it modifies molecules and transports them within the cell. Okay, so it's moving stuff inside the cell. It doesn't ship it out. Now we have two types. We have the rough ER, which is located right here. You'll notice how there is ribosomes attached to it. It gives it a rough-looking texture. Now over here is a smooth ER. No ribosomes are attached. Same thing. You're looking here. Smooth, rough. Okay. Endoplasmic reticular moves stuff within the cell, okay? Now the Golgi apparatus also moves things, but this moves stuff out of the cell. So it moves stuff from inside and ships it outwards, okay? And it does this using vesicles. And vesicles, I always remember this because vesicles sounds like vehicles, okay? And these vehicles are for shipping things out of the cell. Okay, so Golgi apparatus is where the final modification of molecules occur. Okay, and what happens is we do this final modification, we package them in this vesicle and ship them to where they need to go. Lysosomes are like the garbage men of the cell, and they are basically just a digestive sac that helps recycle parts of the cell, okay? So our lysosome, which is located right here, what they do is when there's a dead part of the cell, they actually merge to it, release their digestive enzymes, and they break down those parts so that they can be reused inside of the cell. All right, so these are all some of the major organelles that we have inside of our cell. Now, what we're going to talk about next is transport. So passive transport is just simply the movement of molecules from high concentration to low concentrations without using any energy. So if we notice here, we see how we have a high number of molecules here. What's going to happen is these molecules are going to naturally want to move across until they have reached what is known as equilibrium. Now, equilibrium is when there is an equal amount of molecules on both sides. Now, this movement of molecules from high to low concentration, we call this the concentration gradient. Okay, and the concentration gradient is just this movement from high to low of particles without any energy. So in passive transport, no energy is needed. Okay, no energy is needed to move the molecules. Now we have a couple different types of passive transport. The first one we want to look at is diffusion. And diffusion is the movement of molecules. Okay, so this would be like CO2 or O2. Okay, and these are bigger molecules and they need to be able to move across the membrane. So diffusion, movement of particles from high to low across the cell membrane without using any energy. Now, sometimes these molecules are actually really big and they can't actually pass through the membrane like we see in diffusion here. And this is called facilitated diffusion. In facilitated diffusion, there are these proteins that span between the cell membrane. And these are like channels or tunnels to kind of get through. So what happens is transport proteins, like what you see here in purple, help these ions and polar molecules to diffuse through the cell membrane. It's a lot easier. It gives it a helping hand. Facilitate is like to help. So facilitated diffusion helps these molecules go through the cell membrane. Now, another type of passive transport is osmosis, and this only has to do with water, or H2O, okay? Now, osmosis, the diffusion of, again, water across the cell membrane. Now, we have three types of osmosis, or three types of conditions water can be in. Isotonic, where the water is moving in and out of the cell, and the cell doesn't shrink or gain any size. But we have these two other conditions, hypotonic and hypertonic, okay? I'm only going to write the first couple letters. And really, the only letter we want to look at is the first letter that's different, the O and the E. So in hypotonic, the O is much bigger than the circle in the E. So hypotonic, the cell gets bigger, okay? And in this case, you'll notice this right here. Sometimes when we have a higher concentration of water outside the cell than inside the cell, water wants to go in, and it can actually cause the cell to pop. Now, in hypertonic, the E is smaller, the cell gets smaller, okay? And when the cell gets smaller, we often call this, it often shrinks. So there's a higher concentration of water inside the cell than there is out, and water flows out. Again, hypo and hypertonic. Now, active transport is completely different because energy is needed in order for this process to occur. So this energy is in the form of ATP. So active transport requires energy in the form of ATP and moves molecules against the concentration gradient. So in active transport, we're actually moving molecules from low to high concentration, okay? So against the concentration gradient. And as you can see here, we need ATP, our energy molecule, to perform this process of moving those against the concentration gradient. Now, one of the most, we need to move these molecules out or into the cell. So active transport, again, energy is needed in the form of ATP to do this. So exocytosis is the movement of materials out of the cell, and endocytosis is the movement in the cell. Now, how I always do this is exocytosis sounds like exit. So the molecules are, in this case, leaving the cell, okay? Endocytosis, it sounds like enter, okay? So the molecules are entering into the cell via vesicles. And again, vesicles are like the vehicle of the cell, transports things from place to place. Now, one of the most important active transport mechanism we have is a sodium-potassium pump. So in the sodium-potassium pump, one of the, in nerve cells, the pump is used to generate a gradient of both sodium and potassium ions. And the reason that this occurs is because it's used to transmit signals through off nerves. So what happens here is we're gaining both a high concentration of sodium and potassium. And we do this, okay, so if you look down here, we have 3 sodium molecules, and they bind to this carrier protein. ATP attaches, it changes its shape, okay, and what that does is then the sodium is released and 2 potassium ions come in, okay. So as a net gain, we get 2 potassium ions on the inside and 3 sodiums on the outside. And what this does, it creates an electrical imbalance so that our nerve cells can occur. Again, sodium-potassium pump, moving of both sodium and potassium ions to create a concentration difference. So we're moving these molecules against the gradient. All right? So hopefully this video helps you. Again, this is the cell and cell transport, topic 3 in the Keystone series. Hopefully these videos help you out for the Keystone Biology exam at the end of the year. This is Mr. O'Brien signing off.